Welcome to another deep dive on G Fun Facts Online. Today we are documenting a massive paradigm shift in the history of medicine that has unfolded over the last 12 months. If you follow biotechnology, you know that 2025 marked a definitive turning point, a moment described by oncologists as the time when the drug manufacturing factory moved inside the human body. We are talking about the transition from ex vivo to in vivo CAR T-cell therapy, a technology that effectively turns a patient's own blood into a bioreactor. To understand why this is such a monumental leap, we first have to look at what we are leaving behind. For over a decade, chimeric antigen receptor T-cell therapy, or CAR-T, has been hailed as a living drug. It has been a miracle for certain blood cancers. But the process of making it, the status quo of 2024, was a logistical nightmare. We call this the vein-to-vein bottleneck. In the traditional method, you have to extract immune cells from a patient, freeze them, ship them to a centralized manufacturing facility often halfway across the country, genetically reprogram them in a lab to recognize cancer, multiply them into the millions, freeze them again, and ship them back for reinfusion. This cycle takes 3 to 6 weeks. For a patient with rapidly progressing lymphoma or leukemia, that delay can be, and often has been, fatal. Beyond the time, there is the cost. With price tags exceeding $400,000 per dose, the therapy is financially toxic to healthcare systems, and biologically, there is a major flaw. When you force T-cells to multiply in a plastic dish for weeks, they get tired. By the time they are put back into the patient, they are often exhausted or senescent, meaning they have lost much of their fighting potency before the battle has even begun. Enter the era of in vivo bioreactors. As of early 2026, the paradigm has shifted. We are no longer just manufacturing drugs. We are engineering the patient's own body to become the manufacturing facility. This approach bypasses the need for external laboratories entirely. By injecting a smart delivery vehicle, either a viral vector or a lipid nanoparticle, directly into the bloodstream, we can seek out specific immune cells, reprogram them on the fly, and unleash them against tumors or autoimmune diseases. There is no harvest, no shipping, and no lab wait. The treatment becomes an off-the-shelf injection, accessible at a local community clinic rather than just a specialized academic hospital. The core scientific challenge here has always been specificity. If you inject a genetic modification tool into the blood, how do you ensure it only modifies T-cells and not your heart, liver, or brain cells? The breakthroughs of late 2025 were driven by two competing technologies that solved this problem: viral vectors and lipid nanoparticles. Let's break down the mechanics of each, because they represent two very different philosophies of treatment. First, we have the viral vector, often described as a Trojan horse. This is the approach favored by companies like Umoja Biopharma and Kelonia Therapeutics. They utilize engineered lentiviral vectors. These are viruses that have been stripped of their ability to replicate, so they cannot make you sick, but they retain their ability to insert genetic material into a cell's DNA. The genius lies in the outer surface of the virus. It is pseudotyped or modified with specific binder proteins that only latch onto receptors found on T-cells, such as CD3 or CD8. Once injected, this virus floats past liver cells and heart cells because it simply cannot stick to them. It finds a T-cell, binds to it, enters it, and permanently integrates the CAR gene into the T-cell's genome. That T-cell then expresses the CAR on its surface and begins to multiply. The advantage here is permanence. This creates a self-replicating army of CAR T-cells. A single shot can theoretically provide lifelong protection, known as immune surveillance. The second approach is the lipid nanoparticle, or LNP. This is the stealth messenger. Companies like Capstan Therapeutics use this non-viral approach. It leverages the same underlying tech that powered the COVID-19 mRNA vaccines, but with a significant twist. These are microscopic fat bubbles containing mRNA instructions for the CAR. The surface of the LNP is decorated with antibodies that target T-cells. When the LNP fuses with a T-cell, it releases the mRNA, and the cell's machinery reads that mRNA to produce the CAR protein. The key difference here is that mRNA degrades. It is temporary. This leads to transient expression. The CAR T-cells exist for a few days to wipe out a target and then fade away. This offers safety and tunability. It reduces the risk of long-term side effects like B-cell aplasia, where the patient permanently loses their B cells, or oncogenesis, where the vector itself might accidentally cause cancer. If the patient needs more therapy, you simply give them another dose. While 2024 was a year of preclinical promise, 2025 was the year of clinical reality. We moved from mouse models to human trials, and the data presented in late 2025 electrified the hematology community. Perhaps the most stunning update came from Kelonia Therapeutics. At the American Society of Hematology meeting in December 2025, they presented late-breaking data from their Phase 1 trial of KLN-1010. This is an in vivo gene therapy using a lentiviral vector to generate anti-BCMA CAR cells for multiple myeloma. In the first cohort of patients, KLN-1010 achieved a 100% minimal residual disease, or MRD, negative response. This is the gold standard. It means that after a single injection, no trace of cancer could be found in the bone marrow of these heavily pretreated patients. Even more remarkably, the treatment showed a favorable safety profile with no grade 3 or higher cytokine release syndrome or neurotoxicity. These are the dangerous side effects that have plagued traditional CAR-T. Kelonia effectively demonstrated that you can safely reprogram cells inside a human to wipe out cancer without the need for chemotherapy conditioning or weeks of hospitalization. Another major player, Seattle-based Umoja Biopharma, solidified its position with its Vivo-Vec platform. In September 2025, the FDA granted fast-track designation to their lead candidate, UBVV111, for large B-cell lymphoma. By late 2025, Umoja had initiated patient dosing. Their approach is fascinating because it solves the problem of cells dying out too quickly. Their vector delivers not only the core gene but also a rapamycin-activated cytokine receptor. This is essentially a remote control. It allows doctors to give the patient a simple pill, rapamycin, which activates the receptor and boosts the number of CAR T cells inside the body on command. This tunability was previously impossible with permanent viral vectors. Their confidence was further bolstered by a massive $100 million Series C financing round in January 2025, signaling strong investor belief that the future is indeed in vivo. While Umoja and Kelonia focused on cancer, Capstan Therapeutics took their lipid nanoparticle technology into a new frontier: autoimmune diseases. In conditions like lupus or myasthenia gravis, B cells attack the patient's own body. Traditional CAR-T is often considered too toxic for these chronic conditions. Capstan's transient mRNA approach offers a reset button. It wipes out the bad B cells temporarily to let the immune system reboot, without permanently altering the patient's genetics. In mid-2025, Capstan launched a Phase 1 trial of CPTX-2309. They started with healthy volunteers to prove safety, a conservative approach that highlights just how novel this platform is. Using mRNA to engineer cells in vivo is technically a new class of drug. Preclinical data presented in late 2024 showed deep B-cell depletion in primates, setting the stage for efficacy trials in lupus patients in 2026. We also have to mention Umoja, which is taking a different angle by targeting hematopoietic stem cells, the mother cells in the bone marrow. Instead of just engineering T cells, Umoja's InGeno platform modifies stem cells to produce all types of engineered immune cells, including natural killer cells and macrophages. This is particularly promising for solid tumors, where T cells alone often fail. Engineered macrophages can penetrate the dense fortress of a solid tumor and eat it from the inside. Umoja announced plans to enter the clinic in the second half of 2025, targeting solid tumors that have largely resisted traditional CAR-T. The shift to in vivo is not just about logistics or cost, it is about better biology. There are 3 key biological advantages here. First, we are working with younger cells. Ex vivo manufacturing forces T cells to divide millions of times in a dish, artificially aging them. In vivo engineering modifies fresh T cells inside the body, preserving their stemness and potency. These younger cells are more aggressive and persistent in killing cancer. Second, there is no need for lymphodepletion. Traditional CAR-T requires patients to undergo harsh chemotherapy to make room for the new cells. In vivo therapies like Kelonia's and Capstan's have shown efficacy without this toxic step, preserving the patient's native immune system and reducing susceptibility to infections. And third, specifically with the mRNA platforms, we have an off switch. If a patient experiences severe toxicity, doctors can stop the treatment, and the CAR-T cells will naturally disappear within days. This safety valve is impossible with traditional living drugs that persist for years. However, despite the excitement, the path forward is not without peril. The nightmare scenario for in vivo gene editing is the transduction of the wrong cell. If the vector accidentally engineers a tumor cell instead of a T cell, it could theoretically make the cancer resistant to therapy. While 2025 data suggests high specificity targeting CD3 and CD8, long-term safety monitoring is required to ensure no germline transmission occurs, that is, passing the modification to reproductive cells. There is also the black box of dosing. In a lab, you know exactly how many CAR-T cells you are putting into a patient. In vivo, you inject a vector, and the patient's body produces the cells. The number of cells produced can vary wildly between patients based on their immune health. This variability is why Umoja's rapamycin boost system is so critical. It attempts to control the variable. And finally, there is the risk of cytokine release syndrome. Even without expanding cells outside the body, The rapid activation of T cells inside the patient can cause inflammation. However, early trials suggest this may be milder than ex vivo therapy because the expansion happens more gradually within the body. In conclusion, we are witnessing a semantic and scientific shift. We are moving away from cell therapy, where the cell is the product, to gene therapy, where the vector is the product. By 2030, the complex, expensive clean rooms used to manufacture drugs like Kymriah or Yescarta may look like the mainframe computers of the 1970s, relics of a pioneering but inefficient era. The future of immunotherapy sits in a standard pharmacy freezer, a vial, ready to inject, capable of turning a patient's own blood into a precision bioreactor. As we move through 2026, the results from Kelonia, Umoja, and Capstan will determine if this promise holds. But if the data from late 2025 is any indication, the factory has successfully been relocated. It is now within us.